Great. Thank you so much, Joe. And I want to thank you. I want to thank you and all the folks at the Midwest Glacial Lake Partnership for the invitation, for the chance to talk a little bit about some of the things that we've been doing in our lab group for the past few years, and get some feedback from some folks in the nearby states. Before I jump in, I think this is a point that stands well with the Midwest Glacial Lakes Partnership. You know, science is a team sport, and what I'm talking about today is really I'm sort of the spokesperson and cheerleader for a lot of really great work done by folks I've had the chance to work with, and you will see these names and faces again throughout the talk, because really they have been the ones that have done the heavy lifting. So limnology as a discipline has fairly long roots in the Midwest that go back 150 or so years. There have been some core sets of measurements and things that we've done repeatedly to measure lakes and understand the way they work. And for a lot of these procedures and methods that were developed in the early, the late 1800s, early 1900s, they were robust, and we still rely heavily on these sort of basic approaches for measuring and monitoring lakes. There may be some subtle changes. We don't dress quite as nicely as we used to, but in many ways, some of the basic things we do to study lakes have been persistent. And as this figure here shows, we can look at this picture of Burge and Judet in the black and white photo, who studied Lake Mendota, shown here in this map on the left. And we, through the North Temperate Lakes Long-Term Ecological Research Program, sort of continue with that tradition of Burge and Judet of studying Lake Mendota and other lakes, and have been fortunate enough to build on this and have these tremendously long-term records of a number of variables through the repeated use of these tried and true age-old methods. Now, we have made important forward progress in terms of how we do things, and certainly I think just about everyone is familiar with the substantial step forward that has been possible through the development and use of environmental sensors that can be put into a lake and left there to record data at higher frequencies and develop sort of more detailed temporal understanding of what goes on. This is David Bowie, who now resides in Lake Mendota and is measuring and monitoring things for us when we can't get out there on the lake. And having buoys that are deployed across lakes around the world, we've been able to see things in remarkable detail. We can measure things like temperature, oxygen, chlorophyll, a number of basic water chemistry variables, and develop these lovely, as I said, rich diagrams to show stratification, in this case, of temperature and the deepening of the epilimnion that precedes lake mixing in the fall. And these devices that measure things at high frequencies also give us the opportunity to catch rare events, to see things when we're in the office and not in a boat. So we have this example here of sort of a late summer brief transient stratification that happened within the epilimnion of getting this very warm layer in the top 5 meters of Lake Mendota that persisted there for about a week before it went away. These were things that we often don't catch if we do the sampling manually. And these sorts of measurements have been great. They have really provided us new insight about lake physics and lake chemistry, but they're always done at a buoy at a central location. In this case, here's shown where the buoy is in Lake Mendota. This is the place where these measurements have been taken year in and year out. And the thing that is notable about this particular map that shows Lake Mendota, and it also shows the land use around Lake Mendota, the city of Madison and the farm surrounding it, is we see this great detail about what the landscape is like, but these lakes themselves we just see as blanks, as just water. And so we can really ask ourselves, what exactly are we missing by just sampling at this one point in the lake over time. There's quite a bit more water out there, and we really don't have a feel how representative this particular point is and what we might not be seeing elsewhere in the lake. And yet we know perfectly well, if anyone's been out and spent time in a lake, particularly a lake like Mendota that is eutrophic, is that things are not uniformly distributed around the lake. So here's a picture of a developing algal bloom. We know that they can be patchy and show up in one part of the lake and not another. And this really does raise the question again of what are we not seeing by going to this one particular point over and over again over time. Now, one so critical and high value solution to this has been the growth of the use of remote sensing tools to look at lakes. Landsat satellite imagery has been incredibly valuable for a large number of studies around the world. And here is a picture of one of the famous algal blooms on Lake Erie that showed up. And you can see the actual sort of visual image in the upper left-hand corner, and then a remotely sensed version of this that has quantified the actual amount of chlorophyll that is in Lake Erie. And again, the emphasis here is on these algal blooms and that they are not uniformly distributed across the lake. And so if we have one point in Lake Erie that we're studying, we might think either the lake is all green or the lake water quality is just fine and there really isn't that much going on there, depending on where that sampling point might be. Now these satellites have been incredibly valuable, but they detect what's going on in the lake in a similar way that we do. They rely on visible light signals. And so there are only a handful of attributes of lakes that show up in a visible fashion that can be detected with our eyes or via these satellites. Obviously, algal blooms or chlorophyll is one of them. It's very fortunate. That's a really important attribute to be able to sense remotely. Also dissolved organic matter from terrestrial origins, sometimes referred to as FDOM for fluorescent dissolved organic matter. You can see here at brown water spilling into Lake Michigan from a tributary up around the north side of the basin. And then of course, sediment particles also show up, and we can see those with our eyes, and those can also be detected from satellite imagery or from remotely sensed imagery. But there's quite a bit beyond that that we're not seeing with these tools. And of course, the other deal with the remotely sensed images like this is they work well in lakes like Lake Erie that are big lakes. Small lakes, not so well. They become hard to distinguish from that remote distance. And then, yeah, so satellites are good, but they only address a small slice of what is going on in the lake. Now, you can make up for that by putting buoys in there that have a lot more equipment on them and detect many, many more things. But we have these two extremes of satellites, which can detect a few things in big lakes, and buoys that can detect and measure a lot of things, but only at particular points. And I think this dilemma is again shown well by Lake Erie, where they were having their lovely algal bloom that cost them millions of dollars. And they subsequently have deployed a network of monitoring buoys because of the expense and the danger associated with these algal blooms. And these buoys, you can see in these dots up here, are great, except for you can notice that only one of them is actually picking up the start of this algal bloom. The rest of them seem to be in blue water. So we still have a problem with this network of 10 buoys that they put in, of having a lot of blank spaces and having this sort of hit or miss problem. For anyone who has worked with these buoys, while the prices are dropping a lot, these are still very expensive pieces of equipment to deploy in a lake, and they do need some care and feeding. So putting out 10 buoys, I don't know how much that costs, but I do know it is more than I have to be able to put out on other lakes elsewhere. So the question is, if we are interested in understanding or studying spatial variability in lakes and having an idea of what we might be missing, is there a less expensive way to fill in the blanks between buoys, particularly in smaller lakes or for variables that are not chlorophyll or dissolved organic matter or sediments? So in essence, can we move the buoy around from one place to another and maybe get some more information? Well, I can tell you that dragging a buoy behind a Boston whaler is not a viable way to do it. And so what I want to tell you a little bit about today is how we've tried to fill in the blanks and understand and begin to examine spatial patterns within lakes and rivers. So credit really goes to Luke Loken and John Crawford for making this happen. And they are the original sort of generators of what we call the FLAME, the Fast Limnology Automated Measurement Platform. So critical to start with a good acronym and go from there. So I'm just going to call this the FLAME from here on out. So the FLAME is basically like having a buoy in a box and then driving the box around on a lake. So we pump water into a box. We have a series of control valves and make sure the pressure and the flow is correct. We have the water come to the sensor, and then we pump it out as the boat is moving across the water surface. And that's really, in its simplest form, all that it is. This has gone through a whole bunch of versions and development. This is the original FLAME, and we have an expression in our lab which is, if it's not fast, it's just lame. And this one was, the first version was pretty lame. But you know, Luke and John and later Paul Schramm, another grad student in my group, spent a lot of time tinkering. I paid a lot of bills for strange plumbing fixtures at hardware stores. And the FLAME definitely evolved into something that was a lot more functional and seemed to work and be able to allow us to take sensors around the lake and make a lot of measurements. Still, throughout this first year and a half or two of these guys working on this, it seemed like every time I went to see what they were doing, I would have pictures like this. They were in the workshop, toolboxes were open, the lid of the sensor box was open, and something was being fixed or adjusted. So we were fortunate enough to have an opportunity to work with some actual engineers on this. This is the group of engineers from UW-Madison's Physical Science Lab. And when they are not messing around with limnologists, they make things for physicists. So they have made things for the Hubble telescope. Their big claim to fame is constructing a neutrino detector in the South Pole for physicists. And so I think we are their outreach project, and they had fun messing around with us. And here are a couple of the key engineers in this project who looked at our box of wire and tubing and parts from the hardware store and helped us make it a little bit more enduring and simplified and reliable. So after working with these folks, they built this lovely sort of plan and schematic, complete with the very cool James Bond boat, of exactly what this Fast Limnology Automated Measurement Platform would be like. It involves an intake structure, the water being pumped to a box with the sensors in it, and the data acquisition system. And when all was said and done, Barb Biratella on the left, who became the point engineer for us, delivered our FLAME box. And you can see this is just very nice and tidy and just really much more professional than we were able to do in the basement of the Center for Limnology. And so again, here are the parts: the gas, the intake valve where the water goes into the boat. And this is also coupled with a device that allows us to measure dissolved gases in the water. The box in the middle there contains the sensors and devices for controlling the water flow. We have it painted white to keep the heat down. And all of this gets done in, gets put into what is not the James Bond boat, but in fact was a surplus Boston whaler. But it, you know, we were able to put all the parts we need in this boat and go out and basically drive around lakes and measure things of great spatial density. We have tried a whole bunch of different sensors that involve sort of basic limnological sensors to being able to measure greenhouse gases, measuring nitrate. We have a GPS that is essential for knowing where we are. We've done some more sophisticated things for greenhouse gases having to do with nitrous oxide and methane and so forth. But really the workhorse of this system is the multi-parameter sensor. This is something that you can have on your own for a reasonable price of $20,000 to $24,000, but you get a lot of different variables that you can measure, and then the GPS system, so you know where you are in the lake. And the other critical part of this system is the data handling process of being able to take data streams from a whole bunch of different sensors made by different companies, each with their own idiosyncratic sort of way of recording data into a central software system. And we also have the capacity to see where we are in the lake and to be able to see how the sensors are behaving as we drive. This is incredibly useful. And credit for this goes to Mark Gaylor, who has worked with us. He is just a tremendous capacity, not only to work at that hardware software interface of writing the software that's needed and integrating all these different data streams. So when we're out in the boat doing our thing, you can see these two pictures on the right-hand side that we can actually see the data that's getting recorded. These are not the greatest photos, I apologize, but a little glare from the sun there. But this diagram on the bottom is, oops, sorry, let's go back, is showing chlorophyll and turbidity as the boat is being driven. And then we can also monitor how the sensors are doing, how their status is, how they're, are they behaving themselves within bounds. And this is a great feature because if we're doing this and we see something strange or interesting, we can circle back in real time and go in and take a slightly closer look at what's going on. So this is sort of the essence of all the parts of the flame. And let me show you the flame in action. So this is a lovely drone video here. You can see the boat in the middle that has that flame, the box and the water intake in it, and we drive in a grid pattern across the lake, and we can actually make a GPS trail and follow that. So we do it repeatedly in the same exact way each time. And all sorts of things are being recorded. So the raw data that we get back may look like this. This is the grid with the different concentrations of a particular variable that is being recorded by one of the sensors. We can then take that grid data and interpolate it to get a full 3D, a sort of 2D map of concentration or temperature or whatever the particular variable is. And we can do all of this depending on the size of the lake. This is a small lake that's about, I think this is about 1.5 hectares, and this can be done, in an hour. And we've, as opposed to having just one buoy in the middle of the lake, this interpolated map, as it says here on this slide, is based on 1,000 to 10,000 observations that are distributed across the lake. So in a pretty short period of time, we can generate a great deal of data and really get a sense of what's going on in terms of spatial variability of 7 or 8 different variables. So, when this all started, this was just sort of a little bit of a harebrained idea that John Crawford had, and He thought that there was something in it, that there would be some spatial variability within lakes, that lakes were not well mixed reactors. And again, we had an idea of that was the case in looking at algal blooms in Lake Mendota. But, not all lakes are Lake Mendota. I realize that. So this was one of the early eye-opening trials of using the flame. This is Trout Bog. This is one of our long-term ecological research study lakes. It's about 2.5 acres or so in size. It is in many ways sort of just an unremarkable dystrophic lake, and was one of the first places where these guys really did some detailed mapping. And this is the figure they showed me. Now you can see in this upper left-hand corner the track that they took with the boat. And each of these different grid maps on the grid is a different variable. And there is clearly a great deal of spatial variability in this small little lake that we just did not know existed. And we can see sort of an edge effect, a difference of, excuse me, around the edges relative to the middle. We can see an area in the lower right-hand quadrant of the lake where we're getting high amounts of organic matter coming in, and therefore a lot of carbon dioxide being generated. We can see another part of a lake where we have a lot of dissolved oxygen for some particular reason. And, suddenly this was like an eye-opener for me, that these lakes were just nowhere near as uniform as I thought they were. And I thought, man, these guys they're really onto something. So subsequent to that, Luke and Paul Schramm, and now also David Ortiz, who's a current graduate student, have been taking advantage of the high density of lakes across the northern highlands of Wisconsin to map these lakes and to get a better understanding of what the nature of spatial variability is within any one lake. So doing these surveys of all these different lakes, there's some basic things that come out of this. First, we can look at any one variable, and we can see a range of spatial variation within the lake. So this is work from Paul Schramm's master's thesis. We can look at how much light is penetrating through the water column. And it varies from the extreme example of Little St. Germain Lake, where you have remarkable amounts of variation in terms of light penetration within the lake. So up to 3 meters difference in terms of light penetration in Little St. Germain from one part of the lake to another. To the case of the Manitowish Lake chain example here, where we have a gradient of about 1 meter of light penetration from one side of the lake to the other. And then you have lakes like Diamond Lake that effectively there is no spatial variability. It's all about 8.7 meters of light penetration. And this has earned Diamond Lake, in Paul Schramm's words, to be the most boring lake in the Northern Highlands. We can look within any one lake and we can see that different measurements, different chemical variables, different biological variables vary in different ways. Some are highly variable, some are not. So we can look at pH, for example, that's pretty uniform, or specific conductivity, which is that yellow map, pretty uniform across the entire basin. And then you get into other variables like chlorophyll or methane, which is the upper right there, where you have pockets of variation and you have different patterns for one variable as opposed to another for within any one lake. And we see variation over time. So here is carbon dioxide and methane in Lake Mendota in spring, summer, and fall. And every date has a different pattern to it, and different amounts of these greenhouse gases showing up in the surface water. So I think sort of the big picture is water chemistry and a lot of limnological attributes in lakes is a lot more variable than we knew. And then this has raised sort of the second generation of questions. What are the attributes that are variable? What creates this spatial variability within lakes? What are the ecological consequences of this variation? And what can this tell us about the underlying processes that are going on in any one water body? And is there some things that we can use with this sort of information about spatial variability within a lake or a river? So I'm just going to do in the second part of this talk, sort of a very rapid overview of some of these questions and some of the answers that we have generated since figuring out the technology. Okay, so what attributes are most variable? This probably won't be a surprise. Most of the most variability are those parameters which are sort of chemical or biologically driven factors. The winner of the championship most variability is methane. This is a greenhouse gas that I'm very interested in. Lakes and rivers are great sources of methane to the atmosphere. This is generally a natural process, although it is enhanced by eutrophication. Chlorophyll is pretty variable, and turbidity shows up. And of course, turbidity is a combination of phytoplankton, but also sediments. And then there's some that just really don't very much, like pH and specific conductivity. And we can use these sorts of mapping exercises to basically figure out what the spatial scale of that variability is. So using some sort of spatial statistics, and in this particular case, you can look at the example of fluorescent dissolved organic matter, this sort of brown terrestrially derived stuff that makes water dystrophic. And this is a variable that doesn't show very much variation from place to place. So we sort of say the patch size or the scale of variation for this variable is, on the order of 800 meters to a kilometer. So if you have a small lake, it tends to be much more homogeneous in terms of this variable, as opposed to chlorophyll, where you can see spatial variation on the order of 200 meters or so. So that's actually a very small scale. So if you have a big lake, you can see enormous amounts of variation in chlorophyll over pretty small distances. And then the next question is, well, what creates this variability? And I think, like a lot of ecological questions, the answer is it depends, and it's complicated. But some of these things make sense. And right off the bat, one that's very obvious is sort of landscape position of these lakes. Isolated lakes or headwater lakes tend to be more uniform. We know this. And the ones that tend to be more variable are drainage lakes. The inputs and output tributaries can have a very big effect on that spatial variation. So in this example map shown here, that a lot of that gradient reflects the flow of water through this lake that is on a chain of lakes. So basin shape is another one. This is probably not terribly surprising, that more uniform shaped basins tend to have more uniform water chemistry. So we have that round lake there, that's Crystal Lake, another one of our study lakes. It is actually fairly uniform in its distribution. And think back to the example of Diamond Lake, the very, very boring lake that was very uniform, and that was a pretty square or symmetrical basin, versus the example shown here of Big Muskie Lake, where you get bays and areas where the water doesn't exchange quite as much, and you can really get those areas to go off and have a very distinct chemical identity that is pretty persistent. And then the third factor here is basically how productive the lake is. And this is stuff that David Ortiz is now working on for his PhD, and sort of how nutrients create sort of productivity of lakes, and how that productivity plays out in terms of spatial variability within lakes. And it just seems in general that the more productive the lake, the more spatially variable it is, up to a point. And then if it gets very eutrophic, then everything is just uniformly green or uniformly rich in nutrients. So there's a little bit of a hump shape relationship there. But David will tell us more about this in a couple of years, I guess. So then, another question that we've poked around it is saying, great, there's all this spatial variability. Isn't that nice? So what? What are there some consequences of this spatial variability, or is it just sufficiently small that it's not all that ecologically meaningful? Well, one example where we think there really is some importance to this spatial variability, and the example here, the first example, having to do with benthic primary production. And we know that a lot of lake productivity and support for food webs can be the result of the growth of algae and plants in the littoral zone. We've examined, this is again work done by Paul Schramm, looking at light penetration, or sort of how far down into the water column that light goes. And he did these maps of light attenuation across a whole bunch of lakes, and was able to combine that with lake bathymetry to figure out how much of the benthic sediment was actually receiving enough light to support primary production. So in this map on the right, all these areas in green receive enough light to be able to support, benthic algal growth or benthic plants. And we can actually match that light penetration with some simple models of photosynthesis to get an estimate of that productivity. And this really tells you sort of where there is valuable, I guess, littoral habitat that may support, again, provide support for those food webs. And a lot of times when we do maps and things like this for lakes and trying to make these estimates, we by necessity assume pretty uniform distribution of light or of that 1% depth. And this provides a much more sort of a complex and realistic view of where that light is getting to and where the primary production is being supported on the lake sediments. And then the one that I'm very interested in, this is work done by Luke Loken, is about how lakes, what role lakes play in greenhouse gas emissions. And here again, the carbon dioxide and methane example. Now, carbon dioxide and methane are products of the breakdown of organic matter and microbial respiration in lakes. And we know that most lakes are sources of these gases to the atmosphere. And so, excuse me, Luke did this very detailed mapping of Lake Mendota from spring to fall to figure out how much carbon dioxide and methane were either going into the lake or coming out of the lake. And what he found was a lot of spatial variability in these gas movements in and out of the lake. And so these graphs on the right show each gray line is a different point in the lake, and it is sort of the estimate of how much gas is either moving out of the lake, those are the positive values, or moving into the lake, those are the negative values. So you can see for carbon dioxide in Lake Mendota that CO2 for most of the year on average is being pulled into the lake. It's supporting all those lovely algal blooms that we get. But that's not uniformly the case. There are a lot of parts of the lake where there seems to be carbon dioxide coming out of the lake throughout the year. And then you can see that big kick up in the fall. That's when the lake is turning over and a great deal of carbon dioxide is being sent up from the hypolimnion to the atmosphere. The solid red line and the dashed red line for carbon dioxide, which are right on top of each other, is the dashed line is where the buoy is and what you would guess is going on with this carbon dioxide movement if you just measure things at the buoy. And then the solid red line is if you do the average of across the entire lake based on the maps. And I think what this tells you here is that averages miss a lot of the variation. And for CO2, and the same is also true, actually more so for methane. You can see there's a greater scatter or range in those gray lines, which are those, again, those individual points throughout the lake. And if you quantify this, if you go out and you did this study of gases moving in or out of the lake just based on visiting the buoy, you would end up overestimating the movement of carbon out of the lake by 21% and underestimating the methane by what seems like a small amount, by 8% or almost 9%. But if you just sort of did the difference between what the buoy sees, just sort of the average difference and the spatial difference, it actually is quite a bit worse for methane that you actually can miss by up to closing in on a 40% error if you just go to that one point. So really understanding and measuring these spatial variability can have a substantial effect on what you think the whole ecosystem is doing. So those are some very quick things that we've been looking into. And then, of course, the other side of this, too, that we're interested in is, given that we can generate these maps of water quality and water chemistry pretty quickly, it seems like there should be some opportunities to use this for some important water quality or water chemistry issues. And again, this is well represented by the fact that you can see algal blooms. We can pick up algal blooms pretty quickly with these, faster than the buoy sees them, certainly, because we are covering the whole lake. So some of the things that we've looked at in this context of sort of water quality issues is identifying points, tributary that enter lakes that are particularly noteworthy in terms of what they're contributing to the lake water. So the example on the left is from the UW-Madison campus. And you can see the yellow arrow there shows a tributary that is flowing into one of the bays of Lake Mendota. And below the yellow arrow is a parking lot that holds a lot of the parking for people that work at the hospital or on one particular side of campus. And you can also, this is also a place where the UW grounds deposits their excess snow in winter, and they just let it sit there in this great huge snow pile. And when that snow melts, it tends to drain into that tributary and go into Lake Mendota. So what we're seeing here is the extent of a salt plume that has come in as a result of that melting snow pile off of that parking lot area and the accumulated snow that gets piled up to get it out of the way. So that was a little bit eye-opening to see that this, what is actually a pretty small tributary, can have a pretty big effect on the conductivity status of this part of the lake. Another effort that we have done was using something called the choliminder, which detects the enzyme activity associated with fecal coliform bacteria to develop these flame-based maps and identify tributaries that seem to have high coliform enzyme activity. And this is sort of a very quick and dirty way of suggesting basically, you know, sewage contamination potential that may be entering the lake. And here, you know, the yellow arrows show 3 of the really big peaks in the Lake Mendota, Lake Monona, Lake Wabisa chain. The upper very bright red plume there is from the Yahara River, and this river drains a lot of agricultural land, so we are probably picking up coliform activity from cows and such. But then within the city itself, we can see some other tributaries where that fecal coliform activity may be associated with septic tanks. It may be associated with parks and goose and dog poop. We don't know. But at least it points us in directions of places that we ought to be paying attention to and figure out what's going on with respect to these water quality issues. And of course, I would be remiss talking to this august group not to mention something useful in terms of water quality mapping and having to do with fish. Thanks to Gretchen Hansen for sending me this map at the last minute. But you know, those guys have done some really lovely work to examine lake temperature profiles and highlight where across the Midwestern region lakes may be warming at a particularly fast rate and where this may be getting us into trouble with maintaining cool water fish species, such as our friend the walleye, and honing in on problem regions. And I think the other side of this story is we can use devices like the Flame platform to recognize that within each one of these dots, within each one of these lakes, that there may also be additional heterogeneity in terms of the temperature, and that not only are some lakes not as good as other for walleye, we can get within any one lake that some parts of the lake may be better for walleye than others by looking at these more nuanced and detailed maps for an individual lake. So, this is just the beginning. We really spent a lot of time making this work. And now we're doing the fun part of the science of really understanding spatial variability and what it means for the lake and what we can do to use this to help us manage and understand how lakes work better. And if you have ideas, we're always welcome to do it, and we'll just sign out with the little mapping.
Drinking coffee, sampling.
So thank you very much.
All right. Wow. That was really, really fascinating, Emily. Thank you. I'll let you take a break and grab a glass of water while I express appreciation from the folks that are on the webinar. It's always a little bit anticlimactic when you finish a webinar like this. We wish that we could clap and say thank you, but you'll have to settle for my thanks.
All right.
So we've got, again, the ability to answer and ask questions. Please, if you have questions, use the Q&A function. You can, again, find it by hovering your mouse over the screen and clicking on the Q&A button. Submit your questions through there. We've got a handful that are coming in already, and we'll get to. And I've got a whole list of questions I want to ask as well.
Oh.
So,
Joe, should I stop the share? Sorry.
You can. It doesn't matter.
I'll just. Oops. There we go. We'll end on that one.
There you go. That's a nice slide. So I'll get right into the questions then. First question: does the flame system have the capability of sampling at different depths, or is it just surface sampling? So have you looked at the opportunity or a possibility to put a longer pipe on the end of that thing?
Yep. Yes, and that is the most important question. It's the one probably I get the most. Let me answer it by saying, in the depths of Lake Mendota, there are lots of pieces of sheared off pipe and PVC and things like that were snapped intake valves. So the problem is, if you have a deep intake pipe, that creates drag, and the faster you go, the more likely it is you'll just snap it off. And the idea here is to be fast. And if you're willing to do it slowly, it'll be okay. But because we've been trying to cover larger areas, we have just left it at the surface.
You're trying not to be lame.
Yeah, trying not to be lame.
That's right.
Yeah, and the other side is too, a lot of these lakes, we're not really quite sure what the bathymetry is, and so we don't know if we're going to run in and hit something or run into a bar or something like that, and that's also why there are a piece of sheared off pipes in Lake Mendota as well.
Sure. So this is a question: the time of day and weather would be a variable attribute that might affect what you see and what you map. And so have you seen that in your own implementation of flame? When you go around the lake and then your loops bring you back to the same point, now you've got two different measurements at that same point. So I think that's one part of the question. Then the other part of the question is, if I'm conducting some water quality monitoring and sampling, what advice would you have for me or for somebody else in designing a protocol that might help to accurately represent the lake that we're interested in?
These are good. So yes, the time of day makes a difference. We know that lakes are not constant over 24 hours. The good thing about being able to do the mapping pretty quickly is the idea is to try to constrain that temporal effect. We know it changes over 24 hours, but if we can get the lake done in an hour or two, then the temporal effect should be pretty small, we hope. I will readily admit that for a lake like Mendota, we tend to do the mapping in the morning, simply because the water is calmer in the morning, and that makes it easier. So we are, we're missing something because of that. And we don't have a good handle on what the variation is over the course of 24 hours. And so the question about how does that temporal variation play out spatially is something that David Ortiz, my grad student, is actually working on this summer, and has been doing a series of 24-hour flames where he goes out every 2 hours and maps it. And they just follow the GPS track. He said it's a little weird to be doing this in the pitch dark, but I'm really excited to find out what that's going to look like. Yeah, really good question, and I'm excited to find the answers. As far as when to do the sampling, during what time of day, there is, again, this sort of trade-off between wanting to get smooth water for a lake like Mendota that's kind of big. And I think there's pros and cons depending on what you're really primarily interested in. If you want to go after blue-green blooms, the first thing in the morning is not the best time of day to get them. Sort of midday to afternoon is better. So everything's a trade-off, and you have to decide what the most important thing is and let that be the thing that drives your decision making. That's kind of a lame answer.
All right. Yeah, I think that's about what I was anticipating, though.
Yeah.
It's complicated and it depends.
Yeah.
All right. Next question. This is sort of a Michigan question, but probably applies. So approximately 25% of the inland lakes in Michigan don't turn over and vertically mix completely in the spring each year. And that has a big effect on in-lake nutrient delivery to the photic zone for algae growth during the spring and summer growing season. So could the flame system be used to help understand lake mixing?
I hope so. It should be, well, I'm thinking and talking. I think it can give you again, because we're focused on the surface level, it can tell you bits about the surface variability. I think with respect to those lakes that don't turn over, I know that the ones that I'm familiar with are basically small dystrophic lakes that stratify before there really is a chance to turn over. And I will say for those lakes, I feel like we have learned more about their stratification and thermal profile through deploying temperature strings in different parts of the lake and under ice that give us the depth resolved data than we can if we get just the surface data for these systems. What we do get from these is knowing where you may be getting groundwater coming in, or cooler or warmer things that might affect some of those inputs, though, and that is definitely a strength of this approach.
All right. Do you have any sense for how disruptive the boat's engine would be to epilimnion readings?
Yeah, good. That's the short. Well, we hope not much, right? So the intake is mounted in a front-facing fashion in front of the prop. So we hope that we are not, well, we know actually, because we've done some complicated things with salt additions in front of the boats, things like that, that the water we take in is not being mixed up by the prop. And, I think for a small lake, if you keep going back and forth, you will create some mixing and blurring. But again, the patches of water chemistry differences that we're looking at are on the scales of anywhere from 25 to 200 meters. And I think at that scale, the boat motion is not disrupting that spatial structure. And we've also done it with smaller trawling motors and things like that. And so we feel like we're getting a pretty good, we're not creating the patterns that we're seeing artificially.
Gotcha. Along the same lines, what's the lag time from, at speed from intake to hitting the sensor? How long is that tube, and how does that affect your?
Yeah, so we have to do this thing we do it every year, even though we don't necessarily change the physical structure of the thing, where we measure the time from when water goes into the intake and to when it hits the sensor and gets recorded. So we have this big bucket. We start with a big bucket of brine. We also have another one that's a big bucket of nitrate, so that we stick the thing in there. We know it goes in, and we wait to see it show up on the computer screen. And we make this correction for that lag time, and it differs for each sensor. Some sensors respond quickly, some respond slowly, some are better flame suited than others because they do respond slowly. Like the coliminder, that was a very slow responding sensor. But it takes the water, it does not take the water very long to go from the intake pipe to the box. That's on, that's like seriously, it's like 2 seconds. It's a short line and the water's moving pretty quickly. So.
Similar
but
question
really,
on calibration. Looking at some of the probes that you've got on that list,
Yep.
All of them have their own calibration procedures. Do you essentially just calibrate the probes independently per instructions and then hook them up into the system each time?
We do. Yeah. Yeah. And then, a little bit of a pain, but if you don't calibrate your sensors, you're not going to get good data regardless of how you're using them. So yes, we do. Every time we go out, and like I said, we also check that response time a few times over the course of the field season to make sure it's constant, and we figure that in when we do our maps.
Sure. Okay.
Yeah.
How do scientific breakthroughs that measure spatial variability compare to those of the last century that pioneered in measuring the stratification of lakes?
Oh, wow. Well, we're not. I don't know, I guess, is the short answer. Time will tell. I mean, I think we really are in the early days of developing the ability to generate these maps. And, we're not alone. There are other people who have done this for a few variables or things like that. And then you can buy very expensive pieces of equipment that will map on their own. Well, time will tell, I guess. I think that the lake stratification is a fundamental defining feature of lakes. That's a tough one to overcome in terms of the strength of that understanding. But I do feel like there is a valuable contribution here to understanding the significance of internal processes in lakes and how that heterogeneity how we need to pay attention to that to really understand how the whole lake is functioning.
I think that's fair. Last question I've got here from the audience. So Madison's urban lakes appeared more turbid earlier this year, with cyanobacteria appearing earlier too. So what are the causes of these troubling conditions so early in the year? More of a
That's a
question.
Yeah, that is well, it's going to be those basic things of nutrients and warm water, I guess. It does seem like we got an early jump on a lot of these blue-green blooms. Somebody asked us if we saw any effect of the pandemic on water quality, and hoping that, maybe that was involved. But, most of the blooms that we see in the southern lake, in Mendota and things like that, are tied to weather patterns. I mean, their nutrients are abundant, and so given the abundance of nutrients, it has to do with the spring wetness, it has to do with the spring warming, and so those must be the things this year that really let them get ripping sooner than usual.
Gotcha. Thank you. I've got a few more questions for you, and I'll mention too while I ask those that if folks have any follow-up questions, now's the time to get them in through the Q&A function. So, thinking about fish habitat, and this is the Midwest Glacial Lakes Fish Habitat Partnership, I'm curious whether you've thought about partnering with folks like Wisconsin DNR or other fisheries researchers to see how this might affect fish, and I'm thinking about dissolved oxygen concentrations and where fish might be choosing to live around the lake. There could be a lot of different hypotheses you could test here.
Yeah, I agree. And we have nibbled at the edges of that and not yet really gotten the chance to do that as much as I think we could. There was one sort of preliminary study that we did was on working with Greg Sass. I say this, it was really Paul Schramm, interested in characterizing sort of walleye areas where there are parts of this particular lake that had that nice combination not only of cool temperatures but also of slightly darker water that walleye prefer, and being able to do that pretty quickly and easily by sort of ringing around the lake. And so that's one of the examples of things. And yeah, like you said, DO is another one that we should be able to go and at least quickly find potential trouble spots of low DO areas that may show up in places that you might think would be otherwise nice sort of refuges. So I think there is quite a bit of opportunity there. Again, while I speak fish, I am not a fisheries person, so my bias has been toward carbon and nitrogen stuff. And I think now that we've got this down, that is something we really hope we can do a lot more of.
Gotcha. Yeah, I think that makes sense. And there have been a number of times where I've approached an area where I thought, this looks like a fishy area.
Yeah.
And for whatever reason, there just are no fish there when you're electrofishing or angling. And sometimes it's a head scratcher, and the next bay over or whatever has that same habitat in it, and it looks like the fish are there, and it's why are they there but not here? And so the spatial heterogeneity really seems like a valuable component to add.
Yeah, and this is another tool in the toolkit to ask more questions about why you see those sorts of differences, for sure.
So I'm guessing the answer is no here, but I'm curious whether you've had any interest from some of the commercial and production companies. We see, for example, sonar and side scan and all the different components and technologies that are rapidly expanding now. And I think about the average bass fishing boat that's got just gazillions of dollars of instrumentation on it now that can out map most DNR boats. And I guess I'm curious whether there's been any interest in scaling this up to a commercial aspect, either for resale to other researchers or whether there's interest from the angling community.
Yeah, that's a good question. When these guys started building it, they actually had a discussion with the patent office about patenting this, and we didn't happen. But we have not gotten any of the commercial interest from anglers or fishers on this, but we have had a lot of conversations here and there with folks interested in water quality monitoring. And things just, like everything, got put on the shelf starting around March 2020. And that's so these things have come up. The question is, I don't know. I'm not sure I really want to be a business person managing Flame Inc. or something like that. But yeah, I think that there has been recognition of what this can do for water quality monitoring and doing it really rapidly. Interestingly, one of the contacts I got was from someone from the UN, where this is something you could take into places where they, wanted to do some work in Southeast Asia to do rapid monitoring and looking for sewage inputs for water quality and drinking water purposes. So I think there are lots of that sort of potential for someone if they were more entrepreneurial than I am at the moment. So.
Yes. Sounds like a lot of opportunities for sure.
Yeah.
Last question from our moderator here. In terms of aquatic vegetation management, we see treatments that, depending on how they're implemented, have the potential to kill off a fairly significant amount of biomass in one fell swoop. And so have you, with the Wisconsin DNR or others, had any conversations about trying to monitor or assess DO sags from decomposing aquatic vegetation as a result of treatments or other activities for vegetation management?
I have not, but what a fantastically cool idea to be able to do that. Because we could do the dissolved oxygen and the carbon dioxide and methane as well. No, but wow, now that you say that, Joe, I'm gonna have to poke around and look into that one. I like it.
Yeah,
That would
add
be
it to
pretty
the very
interesting.
long list I'm sure you have. Okay, well, I don't see any additional questions from the audience here. So I'll again say thank you from the MGLP and from the audience. Really a fascinating system that you put together and presentation you've shared. So thank you. I'll remind folks that the recordings of this webinar will be shared. I'll be emailing everybody that registered a link to that. So please share it with folks that you think might be interested, and check out the MGLP website where you can register for more webinars in the future. So with that, thank you. Have a good day, everyone.
Thank you very much.